repeats over here. This sequence,
This meeting is being publicly streamed,
which we refer to as TTAGGGT repeats in human telomeric DNA, this one forms and folds itself and forms a G quadruplex. You can see it's the same single strand which you can see here, which is folding over here and forming quartet, G, G, G. There is a quartet in one plane, then second plane and third plane. It so happens that if you have small molecules or ligands which can bind to it, it will inhibit elongation and therefore it will result in anti-cancer action. This happens under certain conditions, and the strategy is, therefore, a strategy has been developed to have ligands which can bind effectively. Firstly, it should bind effectively. Secondly, it should be able to stabilize this so that it forms a stable complex and it does not let the G quadruplex to open up. So a single strand is required for elongation. So elongation will be inhibited. This is the same thing which I am saying. In other words, this is the thing, and this happens in HTERT in the cellular systems, RNA transcriptase, and so this results into, if the ligand binds, you can see here, and it makes a stable complex, then further elongation will not take place, and it will result into apoptosis, that is cellular death. So what is the basic thing is that you should have G quadruplex DNA and it should be stabilized. To do this, basically, one has to look at first G quadruplex. It's a non-canonical structure in the sense that the normal structure is duplex DNA, which is vaccine and crick DNA. So gonad repeats are there in the sequence, and where have we found it? And this, another feature of this is it's not vaccine and crick DNA. The base pair takes place with the hoopstein seams, which means on the N7 nitrogen atom in the pyrimidine and purine rings. So where is it found? Of course, the first it was discovered at the 3' end of human telomeric DNA. Such tandem repeats were there. Hello? Acha. Okay. Okay. Slide number 4. So where did we find it? Originally we found that 200 base strips, single strands are there, and this maintains the telomeres by telomerase enzyme, and which is overexpressed in cancer cells. In the slide 4, you can see it was found that there are many promoter regions of human proto-oncogenes in which it is found to be there. So that is a very typical one that you know of, maybe c-MYC, c-KIT, BCL2, KRAS, HRAS, which are present in breast cancer, in lung cancer, and in intestinal cancer, cervical cancer. Some of these oncogenes which are present, their promoters contain repeated G like this, like AGG or some other sequence, and GG repeats are there. And it is found that that also forms G quadruplex. Later, it was also found that some viral genomic RNA and bacterial DNA also has repeats. A very typical one that was found was HIV virus. HIV virus has GGG repeats, and there are certain sections and certain genes which are essential for the survival of virus, that they contain G repeats and they form G quadruplex DNA. Similarly, bacterial DNA, I will give you at the end one example tomorrow in the evening class. That is, we have done mycobacterial tuberculosis. So the tuberculosis bacteria which we have, that has got in several genes G repeats. So strategy is being developed. Recently, some people have also found it in malarial parasite. So G repeats have been found in viral genomic, bacteria, in malarial parasite, and so these can become targets. Now these will be the targets. One has to get ligands which can bind to them, which can stabilize them, so that some essential functions are blocked. And once you block the essential functions of bacteria or virus will no longer be able to multiply. In the slide 4, you can see what are these G. I just gave you the sequence. The what do they look like? So this is how they form. It is the what I'm showing you in this particular picture is what we call as unimolecular, which means it's a long sequence which itself is folding. So we see, for example, in this particular A, which I'm showing you, this is the sequence starts from here 5' end, it folds like this, and it folds like this and this. And now you can see this is one plane, this is second plane, and this is the third plane. So there are three planes. These planes are very clear in this particular one. Okay, these are planes. This is the blue structure that you see is gonine. So gonine with gonine, gonine and gonine, and then the second plane and third plane. These structures have been determined by X-ray or NMR, and these are structures HTEL, HTEL22 is human telomeric, and this is c-KIT DNA. It is one of the oncogenes. It's the promoter of one of the oncogenes. Its NMR structure has been done. These structures, if you see, one thing very typical I have shown here, they require sodium and potassium. The reason is, hello? Okay. Every time I will speak. So this is slide number 5. Please see, refer to slide number 5, in which I am showing you some of the single-stranded which have folded. HTEL stands for human telomeric DNA, and c-KIT stands for an oncogene. This oncogene which is there, its promoter has got G repeats. You can see I have shown potassium or sodium here. Reason being, you can see in duplet DNA there are 2 strands. In quadruplex DNA there are 4 strands. So what happens when you have 4 strands in a plane? 4 phosphates in the backbone will come close to each other. In duplet DNA, these 2 phosphates were 180-degree apart. And now since you have 4 phosphates in one plane, this will be 90-degree apart, which essentially means the phosphates come very close to each other. And phosphate negative of the DNA will actually cause a lot of repulsion, and the stable structure will not be formed. That's why it has been found that such structures in vivo, in our system. Slide number 5. अच्छा, माइक्रोफोन ऑफ कर देती हूँ. ठीक है. Now it's okay. So if you see in slide number 5, which I have shown, we need positive ion at the center of the quad. So the quad which we are forming, that is tetrad, 4 gonines that we have. In the center, we need potassium or sodium plus cation as found. These are prevalent in cellular systems. So they will coordinate with O negative charge in the O2 position, O6 position, and then they will try to form a stable complex because of and they will reduce the repulsion. So they coordinate to 8 atoms or 8 oxygen atoms in the purine ring of the gonine. Now another point you can see here, we use potassium. Potassium is available in cellular systems. Potassium fits in nicely. If you put sodium is a smaller atom, this will also fit in. However, there is a difference in the structure. It is found actually by X-ray and NMR. The structure, if you have sodium, is different from the structure that you have in potassium. You can have another alkali atom like cesium and all, but it so happens that is large. It is large in size and it does not fit. So for all our studies, we use potassium and sodium, which are present anyway in the in vivo systems. So often you will see when we talk about this, potassium is higher in concentration. So biological relevant form, if you see, it is in the one which we see in potassium ions. However, in cellular systems, there may be systems where sodium ion is present in excess, and there such structures would be formed. So this is in unimolecular, that means one single long strand of 26 strands for 26 bases, 23 bases, which have folded here into these structures. You can have 4 strands. So this means a short stretch of DNA and exactly similar 4 strands, and they can have G repeats. I will show you in one of the structures. This is tetramolecular in a way because you are having 4 molecules. How do we have it in our system? In biological system, sometimes telomeres pair, which means different DNA strands will pair with each other. These have been found in some recombination, DNA recombination. They have been found to interact with HIV GP120 protein in the case of HIV virus. So they do have some relevance. However, the more common ones are these longer sequences, which is unimolecular. That means a long single strand having G repeats will fold like this, and so on. Another thing you can see is that all these structures, ligand is going to bind to it. So the structures, exact structure that is adopted will depend upon the base sequence. For example, c-kit is a different base sequence. It will depend upon the length, like 22 if you take, or 26. And outside, when we crystallize it or do NMR structure, there is some difference in the structure. Nature of cations, as I said, potassium and sodium give different concentrations. Sometimes concentration makes difference. And also, it is the flanking base. Is it A or T? And which are the starting bases that you have? So these are a lot of factors when you take some short strands and when we try to do the structure. So it will depend upon that. You can see now in slide number 6. In slide number 6, I have summarized what do the ligands recognize. Ligands are essentially small molecules which we can synthesize in lab or which may be available in some way, in plant or animal systems. And these ligands, what we expect is that they will bind to G4 DNA. So which means they recognize it when they bind. Where do they bind? They recognize the loops, which I have shown. They recognize the stacked quad, that is 4 bases, 4 gonines which you have in one plane. They recognize the loops and very intricate conformational aspects. When I say intricate conformational aspects, please refer to the first slide which Dr. Pamita had shown, that when you go to the sugar conformation and glycosidic bond rotation can be either anti or syn, which will take the sugar one way or 180-degree apart. The second is the sugar conformation, which may be C3 prime endo or C2 prime endo. So what does it do actually? C3 prime endo or C2 prime endo will put HOH atoms in deoxyribose sugar in different positions. So this is what I mean by intricate conformational aspects. It will depend upon, do you have anti, syn, what do you have, what are the loops, what are the flanking bases. So ligands will recognize these and they will bind in a very, very specific way. What is the aim? Aim is to maximize the interaction. Basically, you can start with some ligand and then you can go on modifying it, have different substituent groups. And the second thing is we must enhance thermal stability. Supposing it binds and it does not stabilize very much. Just to tell you, the very, very first structure which was done with an anthraquinone analog. And they got 23 degrees centigrade stability, which is very high. I mean, if you have a stability which is 23 degrees centigrade in cellular system, it is above your body temperature, which means it's going to be very, very stable. So the idea is to have good interaction, enhance thermal stability. Another thing is, outside the tumor, it should not be toxic. It should not affect the other DNA in other portion. It should affect only the promoter of oncogene. It should affect only the telomeric DNA, any DNA which you don't want it to elongate. Okay, so only that. It should not affect outside tumor, which fortunately in this case is easier, because outside this, if it is specific to G quadruplex DNA, it will not bind to duplet DNA. So which means automatically it will not affect the normal DNA functioning everywhere in the cellular system or in the body. So it will bind only to G quadruplex DNA. So automatically specificity will arise. Second is, of course, specificity for a particular kind of tumor. Like something which binds to c-kit may not bind well to c-myc or may not bind to human telomeric or bacterial DNA. So specificity should be there so that you can develop a drug which is very specific for that virus or bacteria or human beings. So basically the aim is to do these, and to do this or to achieve this, what we want is we have to do structural analysis. Structural dynamics has to be known. And before we do structure, a simple binding and all, we have to assess whether it binds also or not. So what do? We do structural analysis and we identify what is the binding moiety, what is the interaction at molecular level, and then ultimately we try to modify the ligand, synthesize new compounds. Once we know that these are the interacting groups, so we focus on those. Okay. Now here I'm just giving you example of one of the human telomeric sequence. Typical human sequence is TTAGGG. So this is the hexamer. You can see hexanucleotide sequence TTAGGG. And then again it is the repeat. And again it is the repeat. So it is 6-mer which is repeating itself. And it is found that almost 100-200 repeat may be there in telomere. I'm giving you another example here. C-MYC. C-MYC is an oncoprotein. Oncogene. In this gene, we find 4G here, 3G here, 3G here, 3G here. So this folds. And so this is C-MYC. Normally it is found that 3Gs are there and they give you good stable structures, good G quadruplex DNA. Once in a while, in between, 2 stacks may be there, which means 2G may be there. If you have 4G somewhere, it may give you extra stability. So this is actually a part of the DNA sequence of human telomeric, which I have shown you. This is a part of C-MYC oncogene that I have shown you. Coming to slide number 7. Here, I'm going to show you the techniques, how we can use it. But just to introduce what kind of ligands I'm taking as an example or for my application. So I'm going to tell you apply absorption, fluorescence, circular dichroism, and nuclear magnetic resonance spectroscopy techniques. But the ligands that I've selected for my application and results I'll be showing you is anthracinones. One of the simple reasons, as I told you, the very first structure which was shown was anthraquinone based. It was 2,6. This is 1. This is position number 1 where amino is attached, and this is position number 5 on the ring. The one which was shown historically was they had a substituent group at position 2 and 6. I am showing you 1,5-diamidoanthraquinone. So we have synthesized some of these compounds, and you can see in the 1,5, here is the group that we have synthesized and we have added to the amino group. And here is the same group we have added. So it's a bis compound. We have added the same side chain in this case. So I'll show you some of these which are where we have added diethyl or pipridyl group or morphonyl group. And a similar one was 2,6 compound was which was shown historically, which I told you was found to be quite stable, and there are a lot of studies started. Coming to the slide number 8, this is exactly which I am going to show you some of the results. So I told you the ligand, the only thing I've changed it, all are in 1,5 position here. These ones particularly, we have done others also, and we do have in literature results of many other similar compounds. So the compound that I'm going to show you is 1,5 bis compound, and you can see it is piperidinopropionamide. So this is piperidino group. Here it is morpholino group. Here it is diethyl group which I have added. It was NH2. So HH has been replaced by dimethyl or diethyl, sorry. And this is monoethyl. So these are anthracene. This is the basic compound. All of them have double bond O here, double bond O, and NH here. And only the side chain at the end is different. So I have further in my studies applications which I will show you. I have named them L1, L2, L3, and N4. So you can see the only difference they have is in the side chain. You can see if you have O, it may do extra hydrogen bonding. If you have methyl or ethyl group, this is hydrophobic group, so it will go near hydrophobic region of the DNA. That is the reason for doing some such selection. Going to slide number 9. This is the G quadruplex which is very often taken for analysis. This is human telomeric DNA, which is 26-mer we have taken. That means it is a stretch of 26 bases that we have taken. W stands for wild type, which means the actual human telomeric sequence we have selected. And I told you the repeat is TTAGGG, and then again TTAGGG. So the repeat is TTAGGG, and then the whole thing is repeated. And the second one I have taken is also human telomeric. Only thing is it is truncated. 4 bases on either sides have been removed. There are good enough reasons why we have taken this, and it forms stable G quadruplex. This has been shown by X-ray and NMR both. So in crystallographic structure as well as in solution structure, it forms a good structure. Now you can see one thing. This is done in K, potassium plus, and this is done in sodium plus. Basically, the sequence is the same, but we get very different structure. In sodium, we get what we call as 2+2 basket. Actually, it has anti-parallel. So you can see C starts from A, 1, 1, 2, 3, 4, if you start from here, and then it goes like this to 7, 8, 9. So if it goes, see, if the strand starts from here, 5' end, and starts from here, goes like this and goes like this, so it is anti-parallel. And the next one goes this way, opposite to that. So it is again anti-parallel and anti-parallel. So that's why we call it 2 are parallel and 2 are anti-parallel. Okay? And so this is anti-parallel. In this particular case, glycosidic bond rotation is either anti or syn. It is different in the different cases. And there are 3 G tetrads, you can see, 3 planes. 3 planes in which 4, 4 G are there. One is this 16 and this one. The second plane is here and third plane is here. We call these loops, these are the loops. These are the loops. Normally TT goes on the loops because GGG is going to form the base stacking. Base stacking will be done by GGG. G gives the maximum base stacking and TTA forms the loop everywhere. Here also, and this is called lateral loop. In this particular case, which is the most common one we do not have pure parallel or pure anti-parallel. Okay, we have hybrid structure. This particular is called hybrid form 2, but basically what do we mean by hybrid? It has got 3 parallel strands and 1 anti-parallel. So it is not fully anti-parallel or fully 4 strands are not parallel. This is 3 anti-parallel and 1 anti-parallel. So it's called hybrid structure, 3+1 hybrid. You can see here it shows, if you carefully go by the sequence from 5' end to this is the 3' end, 26th base. And you can see that 3 of them are parallel and 1 of them are anti-parallel. There is one more thing you can note here. See here, the chain is folding. It's exactly folding like this one was folding. If it folds to the adjacent strand, we call it lateral loop. Okay? If it folds diagonally, then we call it diagonal loop. And in one case, it will go like this. It is called propeller twist is there. So it is a twisted sort of form so that it can form strand. So this strand and this strand are parallel. So this is a different kind of loop that we have. These are straightforward, simple loops, lateral loops. So lateral loops, and this is twist actually, which is there in this. So this is the most common form in solution structure has been determined by NMR extensively, and people have worked on this system. It is a very well characterized and a very stable structure. And this is the other structure which has been characterized, and it is present in sodium, which means, what about our in vivo system? In human body, if there is sodium concentrated somewhere, then this structure may be formed. Normally, it is potassium we have in excess and in high concentration in cellular system. So we have this structure. So a lot of people will work on this structure, and I will talk about this. Going to the next slide, slide number 10. The first thing we have used is absorption spectroscopy, which is very straightforward, and I'm sure you all know about it. Oh, it's already 10:40, don't worry. So this is OD epsilon. I'll try to do it fast, and you can ask me questions later on. So OD is epsilon. So what we see is change in wavelength. We try to see on ligand has done this or not. There may be red or blue shift. There will be change in absorption intensity. We can determine stoichiometry, stoichiometry, and we can find out binding constant. In the 11th slide, you can see some of these results. It's similar results, so I don't have to go very much in detail. You can see as the ligand, we are taking a good amount of ligand, and then we are adding drug. and we are looking at the drug peak. Drug absorbs or the ligand absorbs around 480, 490, 500 nanometer. So this is where it is, because DNA does not show any absorption. So our calculation and analysis becomes easier. Otherwise, we have absorption, of course, throughout in the range. But at 260 nm, DNA will also contribute, the ligand will also contribute, because ligand is having anthraquinone ring. But at this 400, 480, 500, 600 nanometer, only drug ligand contributes. So this is, you can see from starting from this, how this is going on. We go on adding, we do 40, 50 experiments, the change drug to nucleic acid ratio, and then you plot change in intensity with respect to DNA concentration or so, and you can see, if you see, let's say at the peak position, the intensity is coming down. This is what is shown here. If you see, it is linear, and somewhere it will show you a sudden change in slope, which means epsilon value has changed, which means another complex is formed. So this gives you some idea about stoichiometry. I will quickly show some more slides before I go to the next technique. So slide number 12 shows you this. To do the data fitting, we have done this equation, which has been used. It's a standard equation, and we can find out epsilon B, epsilon free is known to us, epsilon A is apparent for the complex. We plot this versus this, and we, from slope and intercept, we can determine KB and all. Alternately, many people will use scattered plate plot, which can be used for multiple binding sites. We do have multiple binding sites. It leads to some kind of difficulties sometimes when we have dimer binding. Okay. Next is this is just to show you the result. You can see one type of binding was occurring, then as we increase DNA, another type of binding is occurring. So which means two types of associations may occur. If you see lambda shift has suddenly occurred at this. Suddenly so much of lambda shift has occurred, which means a new complex is being formed, and this gives you an idea about what is the stoichiometry. Going to the next slide, 14, this is with sodium. The result, actual slide for, if you compare with what we got with potassium, are different. So going to slide number 15, this is fluorescence. In exactly similar way, what we are doing is fluorescence spectra of the molecule. So of the ligand molecule. So we are taking ligand molecule and we are going on adding DNA. So we are doing titration. In some cases, you can see we got shift of 30 nanometer or 38 nanometer or so on. Okay, this is another result with sodium, similar results. But the idea is, how do we do? The main issue is, how do we do analysis? So going to slide number 17, this gives you an idea. I think in the fluorescence spectroscopy yesterday, when you saw, many speakers told you about that. This is quenching, which means we are adding DNA to the drug. And so what is the rate of quenching we try to determine? We have static quenching, which means it is temperature dependent. It is accompanied by change in absorbance, and there is no change in lifetime. You can always have dynamic quenching. Quenching means reducing the fluorescence. So if fluorescence gets reduced when we add DNA, it could have been that it could be simply what you call as striking the DNA molecule and decreasing. So it could just be an interaction of that kind. So which means colliding with each other. It could have been just a collision. However, if it is collision, it will be temperature dependent. There won't be a change in absorbance and so on. You can have static and dynamic, both kind of quenching in some cases. In our cases, we were getting absorbance changes, and our quenching was basically we were getting all the changes because of binding. Not a simple collision. It was not simple collision. So the molecule was not getting de-excited because of simple collision. It was getting de-excited because it is binding to the DNA molecule. Going to slide number 18, this is another analysis that we use, log equation, which is a standard equation you will find in many books, so that we can determine this. This is another technique which we use because I showed you we got two types of binding together. Multiple binding sites are there. So if you have multiple binding sites, you can do non-linear fitting. You will not get a straight line, so non-linear fitting can be done using origin we use, and we used a particular method given in this particular case. So it is a non-linear curve fitting which has been done to determine what are the KB1, KB2, KB3. You may have 1, 2, 3, 4 type of association. Going to slide number 20. Slide number 20 shows you how this is the log plot, this is turn format plot, and this is also the Sturm-Hermer plot, and this is telling you that there is a see here sudden change in fluorescence. It was increasing and then it started decreasing, and it happened at drug to nucleic acid 2, which means 2 drug molecules were binding to the nucleic acid. So it is giving us an idea of stoichiometry, and this gives us an idea of binding constant. This is slide number 20 using the equations which I showed you. Going to the next slide, this is, I'm telling you now one more thing, stoichiometry can be done by continuous variation. This is a very standard job plot you would have come across, job plot technique, and that is that it is how to do it. This is called continuous variation analysis, in which we take, the only thing you have to do is, you have to take the DNA and your drug fixed concentration. The total concentration has to be fixed. So you start, let's say you start with 0 DNA, then you add 10% DNA, 20%. Then if you add 20% DNA, 80% will be drug. So, but the total concentration we kept fixed. This is called job plot. And using mitosentron, another drug, I will just show you here. This is the result of job plot. If you do job plot, it will exactly give you the stoichiometry. So this is giving stoichiometry. 0.8 means 80% of drug and 20% of DNA. So with this, what we got, 80% and 20% means 4:1 stoichiometry. And in this case, we got at 66% only, which means 2:2. So this means with this sequence, we got 2:1 binding. With this sequence, we got 4:2 binding. And we have used a tetramolecular DNA in this particular case. So this is job plot which can be used. Going to the next slide, that is slide number 25. This is fluorescence lifetime we have done. Since there are more than one component, one is simply bound and one is free. So both of them will contribute. So you use this kind of standard method and try to determine what is the fluorescence lifetime. So use this has been done, and this is the plot. You can see in the plot, which is slide number 27, shows you the plot. 26 and 27 show you the plot. You can see there is not much change on binding. So, but once you do the analysis and find out, see, this is free drug and this is this. And you can see it's not much, 32.27.2 this is in, I think this is in nanoseconds, and this is this. 2-3 components are there, and you can see that there is not much change in the lifetime. So which means we don't have dynamic quenching, we had static quenching due to the binding. Going to slide number 29, this is the next technique, which is structural technique. And I will tell you about circular dichroism spectroscopy. This is, so far, you have learned about absorption and fluorescence. They are all using unpolarized light. The reason is, when you have let's say, a xenon lamp or a tungsten lamp, all molecules are emitting, the molecules are emitting light on their own. There is no phase coherence between them, and each is independently emitting in all kinds of directions. So light is an electromagnetic field. So if you have an electric field in one direction, let us say in this direction, then magnetic field is perpendicular to that, and the propagation is in this. Electromagnetic radiations are transverse waves. Transverse wave means the propagation, supposing the light is going in this direction, in Z direction, then the XY plane is the plane in which electrical and magnetic oscillations will exist. So this is, let us say, XY plane, electric field and magnetic field, and the propagation is in Z direction. This is a typical light that we have, electromagnetic radiation. So this particular which I am showing you is linearly polarized. The reason being, see here, electric field is going increasing, decreasing, increasing. It is sinusoidal wave. Okay. So the amplitude is increasing, decreasing, increasing. So if you plot this part of amplitude, basically this amplitude is going up and down like this. It is taking a straight line, so it's called linearly polarized light. What source of light, like xenon? Xenon is used normally in some instruments. Earlier we were using tungsten lamp. They all emit unpolarized light. When we let it pass through a device which we call as polarizer, it's a particular kind of crystal that we have. It will absorb light, electric field. Their electric field is on all sorts of direction except one particular selected one. This selected is according to the crystal that we orient. So this is a crystal that we have, which we call this system as polarizer. So we use normal lamps we use, xenon lamp is used, then we place a polarizer in front of that, and what we get is linearly polarized light. Just next to that, if there are some phase differences and all, you can have light, see here, amplitude is changing. In this case, amplitude is not changing, but the electric field vector, the tip of the vector is circular. It is progressing in this direction, but it is circular. This is called circular polarized light. If you have light, this circle going in clockwise direction, we call it right-handed circle. If this goes in anti-clockwise direction, this vector, we call it left-handed polarized light. So we use LS light and RS light. How do we get it? Once again, actually we have to create some kind of phase difference between the light and some combination plates we use, and we can get LS light, we can get RH light, we can get linearly polarized light. What is the basically technique? This technique that we have is the CD spectra is nothing but it is the difference in absorption of LS light and RH light. If you have a symmetrical molecule, use any molecule, a simple conjugated ring or a benzene ring or something like that, if it is symmetrical. So if it is symmetrical, it will not give you CD spectra, in the sense that it will absorb LS light to the same extent as RH light. If the light is absorbed LH and RH in the same way, which will happen in symmetrical molecules, so then it's not going to give any CD spectra. So what is CD spectra? Circular dichroism spectra. CD spectra is actually nothing but OD or absorption. So I will use the word OD, or I will use somewhere in my equation A. Capital A is what we use for absorbance. So it is basically AL minus AR, which means absorbance of LH light and absorbance of RH light, they are different. Why? Because we have asymmetry in molecules. This arises, CD spectra is only and only. So what it means is, and what wavelength will you observe? For example, DNA absorbs at 260 nanometer. OD we will get at 260. CD also will be at 260. Basically, it is absorption spectra. It's nothing but absorption spectra, but instead of using unpolarized light, we will use LH light or RH light. And we will find the LH minus RH difference. What is the difference? This difference is, by the way, very, very small. If your OD is 0.5, it will be 0.0005. It is 1 by 1000, which also means you must have very good source. You must have very good source of light. You must have very efficient detector. Otherwise, how will you detect? You all do experiment of OD absorbance, and you can see what you measure. You measure OD of 0.1, 1, 1.5. And your CD value, the absorption is going to be 0.0001. So it's a very sensitive technique, very specialized source and detectors are used, and their efficiency is very high. And it is nothing but absorbance LH minus absorbance RH. And it comes from asymmetry. Why is it important to us? Going to the next slide, slide number 30. See, why is it important to us is, look at our DNA molecule. It's a small molecule and around one axis. Okay, look at your protein. Protein is alpha helix, it's helical and on one particular axis. So along particular axis all are there. All your molecules, all your atoms are in one particular axis. It is highly asymmetric molecule. So our DNA and protein give you CD spectrum. Just a quick way to understand what is CD spectra, how it arises. You have R light, which means this light is going in right hand, like clockwise direction. You have L light. L light means vector goes in counterclockwise, anti-clockwise direction. If you mix both of them, you can see by vector addition, it will give you linearly polarized light. So if your sample is not there and AR is equal to AR, both are absorbed equally, no CD will be there. So there is no CD. If you put AL light or AR light, if your molecule is symmetric, it will not give you any CD. AL is equal to AR. But if you pass this now, light, if you pass, let it pass through a sample. After it has passed through, let's say, a DNA or a protein sample or any molecule which is asymmetric. All your amino acids, by the way, are asymmetric except glycine. All are asymmetric. So if you let it pass through an asymmetric system, what happens is AL and AR there is some definite value. It is non-zero. It is not zero after it has passed through a sample. Non-zero means, see the blue color. Blue and red were equal. Blue is absorbed, but red is absorbed much more. So its amplitude has decreased. So amplitude of red has decreased. Blue is less absorbed, R is more absorbed. So amplitude has changed. Secondly, blue has gone only this much and red has gone so much on this side. You can see both had gone equal on this side. So this is phase. We are talking of phase difference. So there is a phase difference. As a result, what happens is now you don't have linear light. You have elliptical sort of light and it is tilted ellipse. This tilt is called optical rotation. Many of you, I think in your 10th class or something in your school time, you would have done that cane sugar does optical rotation. This is exactly that. So cane sugar has asymmetric C alpha. So this is the plane of polarization which has got rotated. Secondly, what happens is AL and AR is not equal. Third thing is it has resulted into ellipticity, which is actually minor axis by major axis. Also, refractive index of LS light and RF light are not different. So there are four phenomena occur. They are all interconnected. Going to the slide number 31. So what we do is, for biological purposes and for these interactions, we are using CD. You can do OR, optical rotation measurement. You can do optics. People do ellipticity measurement. Those who have refractometer, they determine refractive index. We will be talking about only exclusively CD, which is simply ODL minus ODR. Going to slide number 31, this is the instrument. See, there is a lot of similarity with absorption spectroscopy. This is xenon lamp normally. This is a monochromator. We use prism or diffraction grating there in our absorption spectrophotometer. It also has polarizer, which I told you. So this gives you linearly polarized light. Then we have some kind of modulator. This is a kind of electroelastic modulator with electric fields and all. We create some kind of phase difference. Once you create that, you will get right-handed circularly polarized light and left-handed. Both can be done, but we alternate. Once we throw LS light, once we throw RS light. So this is your sample cell. Exactly same cuvette is used. We use small amount of sample. This is the same cuvette as you use in OD. So this is your sample, and then there is a detector. Once again, your detector, when it comes out, you can show here it is shown. RCP is big, LCP, both are equal. When it comes out, LCP is smaller, RCP is bigger, which means left-handed got more absorbed. So this is a detector, which is again a very sophisticated photomultiplier tube. Also, we use some charge coupled dual. So the extra thing that you have from absorption spectroscopy is polarizer and this photoelectric modulator. Otherwise, same xenon source, detector, and your cells. This is the instrument. Going to slide number 32. This I will quickly tell you. See, this is CD spectra, and because of peptide bond and pi star transition and pi pi star transition, we get different CD spectra. This particular one is because of alpha helix. This is beta sheet. This is beta turn, and the red one, green one, is random coil. Which means, see, if you take OD will give you some value for this protein. So whether you have alpha helix or beta sheet, it will show you so much of protein. Say, let's say I get OD value of 0.7. So I have this much of protein with me. It will not tell you whether this your protein is alpha helix or so. The same you put in CD instrument. If you get this curve, it means your protein is primarily alpha helix. If you get this curve or if you get this curve, it is this. For example, you had alpha helix and you heat it. If you heat alpha helix, it will become like random coil. So this peak, if you are observing at this wavelength, the value of CD will come down. It will come down and it means you got random coil. Cool it once again, it may become alpha helix. So CD is absorption spectroscopy. It will tell you the structure. Whereas OD will not give you structure. OD only tells you have this much. Whether you have alpha helix or beta sheet, it will not distinguish. Coming to the DNA. In DNA, what you have is, this is the duplet DNA. It gives you a peak. This is the one which we see, this negative peak here and a positive peak here. However, now you look at quadruplex DNA. This is 3+1 hybrid DNA, which I told you, human telomeric, and this is in sodium. This is antiparallel 2+2. So this is 3+1 structure which I showed you, and this is 2+1 structure which I showed you, red color. So CD spectra is very different. If you take OD of this, OD will come out to be the same. Because if your use is let's say 1 millimolar DNA, then both are 1 millimolar. It will give you the quantity. CD will tell you, do you have 3+1 structure? Do you have 2+2 structure? Or is your DNA duplet or single strand? Going to slide number 334, how we use it. So I will just tell you what we do. We look at, this is again a titration we have done. We look at how intensity has changed. We look at if wavelength has shifted, change in structure, or conformation has occurred. Supposing it has gone from 3+1 to 2+1, we get changes, so we get some idea of stoichiometry. Another very great thing we get is we have something like your ligand may have induced extra ligand, supposing it was symmetric, by binding it may have gone into an asymmetric sort of state. If that is the case, you will get induced CD band. Now, theory tells you that if you have positive CD band, large positive in duplet DNA, you have groove binding. If you have intercalation, then you will get small negative peak. And if you have a dimer which is binding, then you will get one positive, one negative peak. Okay, but rules in quadruplex DNA have not been calculated very well. Theory is not so well defined. This is a case which I am telling you. This is mitochondrion binding to human, and we got negative. But actually, we did not have intercalation. We had slight base stacking, which means my ligand was stacking little bit with G bases. So we got deuce. So black shows you when no drug was added. DNA spectra when no drug was added. The moment we add mitochondrion is the molecule, our ligand. When we went on adding, then it gave a negative peak. This, if you get this kind of induced CD band, this is no band, it is zero actually, and this is a band which you are getting a negative band. If you get a band, whether it's positive or negative, the first thing is binding has taken place. Binding has taken place, otherwise how did you get? And binding has taken place in such a way, asymmetric binding has taken place. Your molecule, your drug has become in asymmetric sort of position. So this is negative band which we got. Going to the next slide. Next slide, slide number 35. This is 37, 35. You can see we got bisignate peaks in some other case. This is slide number 36. We did not get, see here flat, here flat, here. We did not get any induced band. So which means drug was not going in asymmetric way. However, if you expand this portion, you can see there was some changes, and this is some changes. So this is 3+1, this is 2+1. So we monitor what are the changes, try to find a binding constant, try to find stoichiometry, try to see if wavelength has shifted. There is no major shift here. Only thing is this shoulder is going down here, which means 3+1 is decreasing, and probably it is going towards 2+2 structure. This is all that we can see. In this case, we did not get induced CD band. So which means our ligand was still in symmetric. Going to next slide, number 37. These are induced CD bands we got in some other example I'm showing you. And lastly, I will tell you is real-time binding. It's a label-free technique. It's a latest technique. It has come 5-10 years back. In this technique, it's a real-time binding experiment. We have a metal film over here, which is actually silver or gold is used. In most cases, we are using a gold thin film. You have a light which gets reflected over here. It is total internal reflection. On this, we put our ligand. Ligand in this case, there we refer to a word as ligand, but actually it is DNA. So we derive this on the gold. We stick G4 DNA. There is a way chemically in which we can stay. So I will stick in potassium environment. So I will get my 3+1 hybrid form. So DNA is sticking to it. Once it is sticking, it is forever. Your chip is gone. This is a chip, gold chip that we are having, and the chip is gone, and we have G4 attached to it. Now what we do is we flow ligand solution, different drugs, and we see whether it's binding or not. So it's a real-time binding experiment. Going to 39 slide. I think I have only 1 or 2 left. Time is going to be over also. So this is incident light, this is reflected light, this is a detector of light like photomultiplier tube, a kind of prism here. This is gold thin film and a glass support. And so light goes here and gets reflected. Now what's happening is, this is the way we have attached here. These, which you can see here, this one, this one, this one, this is DNA. So what you are calling ligand here is DNA. So we attach permanently our DNA. Now we float what we call as annihilate. Annihilate is our drug molecules and small molecules. For example, I will throw one kind of anthraquinone or another drug or another molecule. When we throw it, something may bind, something may not bind. 100% binding is not there. All molecules don't bind. So if it binds, the technique is so sensitive that reflected light will change the angle. So it is the angle that it is detecting, which we call as output RU unit, this reflection that we see. So more is the binding, more it is. I will show you a final slide, one of the example that is in slide number 40. Here. This is the surface plasma, sorry. Slide number 40 and 41, these are the last slides, in fact. So this is what you can see. With H1222, we have added L1 and L2. So what's happening? So actually first DNA is fixed on the slide, on the gold thin film. Now we flow the solution. And the solution we are flowing is L1, which I showed you. This is different concentration. So what we are doing is like putting 1 micromolar, then 2 micromolar, and then 3 micromolar. So what happens is, it binds. These are, let's say, 20 experiments. So in one experiment, we add 1 micromolar, association takes place, steady state comes. So this is the response. The angle has changed, response is there, this is steady state has reached. After that, what will happen? Now we won't flow our annihilate. What we will flow is, we will flow buffer from it. So we dissociate. So this is association and this is dissociation. Next experiment, this is first experiment. Next experiment, we will put 1 micromolar. Then we will put 2 micromolar. So with different concentrations we use, we do 10 experiments or so. So these are several experiments at different concentrations. This is the highest concentration that we have used here, about 5 micromolar. So this is binding has taken place. Here, binding has taken place. Steady state is reached when you continuously keep flowing. So steady state has reached. So reflection angle has changed. So this is the angle, new angle. The change is from here to here. So it has changed, and now you flow buffer. So it will dissociate. So if you see this value, the steady state value, which we call as response unit, and plot it with respect to concentration, fit into the data, it directly gives you real time binding. So this is the binding constant which we determine, and we can determine binding constant from association, from dissociation, and from steady state values. Next slide shows you the same, but it's another example. Oh, I'm sorry, there is something wrong with it. This is the one which I want to show. This is 42 number slide. Please skip 41. Okay, yes, 41. I'm sorry. 41 gives you some results of our experiments. You can see another one. This is the last slide which I'm showing you, binding. So once again, it's the same thing. We see with time what's happening. When you throw it initially one concentration, steady state is reached. We keep waiting, steady state is reached. Now dissociation starts, flow the buffer. Again, take another concentration, flow through it, and this is second experiment, and then dissociate. And then find out from this dissociation, association, as well as from, these are steady state values which have been plotted, this, and get your KB value, or dissociation constant or association constant. So this is a binding, a novel technique which has come up 5-10 years back, whether this only a binding and association like absorption spectroscopy. In the second lecture in the afternoon, I will tell you about other techniques which are used for determining structure of the complex, and then I will show you some mixed ideas as to what is the scope of this work, what else can be done. Thank you very much for listening patiently. If there are I'm sorry it has taken little over 11 p.m. However, if you have questions, you can ask now, or you can ask in the afternoon session, in the second session. You're welcome. Thank you once again.
So, any questions and queries?
I'll wait if there are some questions. Else, since it's already late and we have taken 10 more minutes,
Yes,
you can ask questions. You're free to ask questions in the second
we can have a
session.
question.
That is the
We can
second
have a
lecture that
question.
I'll be taking in the late in the afternoon.
Yeah, late in the second half we'll have questions, right? Okay, Simran, please conclude.
Thank you so much, ma'am, for the talk. It was really nice and informative. I hope you all liked the talk. You are very insightful with it. So we will meet once again at 2 p.m. for the talk by Dr. Aliza. So see you at 2 p.m. Thank you.
Thank you, ma'am.
Thank you, ma'am, and thank you everyone for listening the talk.